Hi guys, welcome to Simple Chemistry. This is the part 4 of the glycolysis series. In previous part, that is part 3, we learned how to write the 10 steps of glycolysis. We started with the blueprint or a short conceptual trick to remember the steps and then went about writing and understanding each step with the structures. In case you have not seen the previous video, I'll leave a link in the description box. Please go and watch it. In this part, we will learn about the enzymes and bioenergetics of glycolysis. You must know our body is a non-stop factory and its tirelessly working workers are the enzymes. All the biochemical reactions happening inside is brought about by the enzymes. Therefore, any biochemical pathway is incomplete without mentioning the enzymes involved. But we need to learn them as well. So let's come back to the blueprint we prepared. Now for the enzymes, there are certain facts. They are divided into categories and usually named as the substrate they are acting on, followed by the action they are performing in suffix. Not always, but in most cases, and here in the enzymes involved in glycolysis, we can use the trick. For type 1, that is ATP exchange reactions, we name the enzymes as substrate plus kinase as suffix. Step 1. The substrate is glucose, but the enzyme involved is general and acts on other 6 carbon sugars as well. So collectively, the substrate here is called as hexose. So the enzyme name is hexose kinase. Sometimes enzymes also need assistance from other organic and inorganic substances. One such category of substances under enzyme terminology is called cofactors. It generally represents the inorganic substances. So here with enzyme kinase, Mg2+, that is magnesium ion, acts as the cofactor. So wherever we mention kinase, we also need to write Mg2+ because without it the reaction cannot occur. Step 3. Substrate is fructose 6-phosphate. Also here, while naming enzymes, we generally use only generic name of the substrate and avoid specificities like numbers and positions. So enzyme becomes phosphofructokinase. And let's not forget the Mg2+. Step 7. Substrate is 1,3-bisphosphoglycerate. So enzyme is phosphoglycerate kinase with Mg++. Step 10. Substrate is phosphoenol pyruvate. Here we need to see another fact about enzymes. An enzyme catalyzing a reversible reaction can be used in both the directions. Since this one is a type of reversible reaction, both phosphoenol pyruvate and pyruvate can be substrates. Since enol pyruvate is unstable, we use pyruvate in the name of the enzyme. Thus, the enzyme is pyruvate kinase with Mg++. Now, for type 2 reactions, that is isomerization reactions, we use substrate plus suffix as isomerase and mutase. For reaction 2, substrate is glucose 6-phosphate. Again, like in step 1, we used hexose because enzyme is common to many 6 carbon sugars. Even here, the enzyme name takes phosphohexose isomerase. In step 5, the substrate is dihydroxyacetone phosphate, which is a 3 carbon sugar, and the 3 carbon sugars are called triose, like 6 carbon sugars are called hexose. And enzyme being common again, the name is phosphotriose isomerase. Step 8. The substrate is 3-phosphoglycerate. Here we use mutase as suffix. So the name of the enzyme is phospho glycerate mutase. Now, for special reaction steps, that is 4, 6, and 9, the enzyme name will be according to the reactions taking place. Now, carefully look at step 4. Here, a compound containing carbonyl and hydroxyls is split into two carbonyl compounds. Now, look at the same step in the backward direction. Two carbonyl compounds combining to form a hydroxycarbonyl compound. This is nothing but the reaction called aldol condensation. Since this is a reversible step, enzyme can take the name with respect to any direction. So the enzyme name here is aldolase. Step 6 is oxidation via removal of hydrogen. So the enzyme takes the suffix accordingly as dehydrogenase. So the complete enzyme name is glyceraldehyde phosphate dehydrogenase. Also, this is extra. Whenever you see coenzymes like NAD+, the enzyme is generally dehydrogenase. Lastly, step 9, enolization. So the enzyme name is enolase. Also, you must have noticed by now, enzyme names usually ends with ase. It is very simple. Just identify the process and it is very easy to recall the enzyme. That completes our glycolysis pathway. And now, let's move on to the bioenergetics of glycolysis. Bioenergetics means nothing but accounts. Accounts of the currency which operates inside our body. Energy currency of our body is the energy compounds. The most popular one is ATP, adenosine triphosphate. So here in bioenergetics of glycolysis, we will see the net gain or loss of ATP. In glycolysis, two energy currency compounds are produced. One is ATP and another is NADH2+. Why is NADH2+ also the energy currency compound? Because it undergoes a process called oxidative phosphorylation and converts back to NAD, and process releases 2.5 moles of ATP. So virtually 1 mole of NADH2+ is equivalent to 2.5 moles of ATP, but in specific conditions only. We will learn about these in few minutes. First, let's see accounts of direct ATP in the process of glycolysis. We will see about NADH2+ later. So in step 1, 1 ATP was used, so minus 1 ATP. In step 3 again, 1 ATP was used, so minus 1 ATP. In step 7, 2 moles of ATP were released, so plus 2 ATP. Similarly, in step 10, 2 moles of ATP is released, so plus 2 ATP. Doing the maths, we get the net gain of plus 2 moles of direct ATP. Second energy compound, NADH2+, if you remember, is produced in the oxidation step where it takes up hydrogen. This was the 6th step. So in 6th step, 2 moles of NADH2 are produced. Thus, net gain in glycolysis is 2 moles of ATP and 2 moles of NADH2+. You should note few things. That not all organisms breathe in oxygen and break down substances using it to release energy. Many organisms, in fact, do the same without oxygen. Point 2. Glycolysis is a common pathway to both types of respirations, that is aerobic and anaerobic. But the fate of NADH2+ depends on whether the conditions are aerobic or anaerobic. NADH2+ produces 2.5 moles of ATP through oxidative phosphorylation, that is, in presence of oxygen, aerobic conditions.
So, in aerobic conditions, 2NADH2 plus yields 2.5 into 2, that is 5 moles of ATP. So as to conclude, in aerobic conditions, glycolysis yields in all 7 moles of ATP, and in anaerobic conditions, only 2 moles of ATP. This briefly completes our topic glycolysis. I hope these videos are helpful for you guys. A very big thanks for watching. If you liked the content, please give it a thumbs up and share it with others. And if you want more videos like this and better, please comment down below these topics you want to see videos on, plus suggestions for improvement are always welcome. Lastly, subscribe to join the league. I'll see you next time. Till then, stay healthy, stay consistent in your work.